Welcome to SB8. Let's dive straight into session number 1, genome writing and synthetic cells. We have 5 amazing speakers in this session covering all the state-of-the-art technologies in the spaces of synthetic cells and genome writing. We have Jason Chin, we have Patrick Kai, we have Julius Fredens, we have Petra, Julius, and we have Kate Adamala. So please join me welcoming all 5 speakers right now, please.
Good morning. My name is Patrick Kai. I'm from Manchester. I would like to tell you, how we can program the smallest unit of life to address some of the biggest challenges the planet is facing today. So there's a paper about 25 years ago when I was a freshman studying computer engineering in China. So the title was, Can a biology fix a radio? So the idea was proposed by Yuri Labunets. So he basically asked the question, when a biologist has a broken radio, if he ever going to approach it to fix it, so how would a biologist approach to fix the radio? He said the first thing he would do would be he would secure funding to buy a lot of identical radios and compare with the broken one. And then somehow he would find a way to open up the radio and look at all the parts inside and solve it based on their size or the colors, and then catalog all of these components into families. And this sounds very familiar to biologists sitting here, right? So this is what we have been doing. We have been sequencing genomes. We have been identifying elements in the genome. Then the next thing we would do is we are going to take out a component at a time. You're going to knock out one component at a time in the radio. If somehow you create a malfunction of the radio or the phenotype of the radio, you will be so excited that actually this particular part may have something to do with the broken radio. And this is what we have been doing. We have been knocking out genes one at a time, and we try to study the phenotype. And finally, he will find a way to bombard the radio and bombard it into pieces, and then he can find the parts which are still connected with each other so that he can identify interactions. And based on these interactions, he can hopefully find out the relationship with these parts. And this is what we have been doing, at least in these genetics areas, identify the interactions. But then he concluded, by doing so, the biologist were never able to fix the radio because we don't really have the mindset of being an engineer, right? So this is what he concluded. Biologists fail to understand the technology systems because they lack the cognition concept of engineers. But at the same time, biology system can be seen as a technical system. And finally, he argued, maybe we should embrace the engineering approach in dissecting the biology systems. So this was 2002. And that was kind of the same time, the inception of synthetic biology. Right? So in early 2000, there are 2 landmark papers, one from Jim Collins' lab, showing you can do a toggle switch, right? So 2 genes replace each other in turns. And then Michael Alloway's lab show you can actually build reprislators to let the biology system oscillate. So this is kind of the transistor moment for synthetic biology. We start recognizing, we can really try to build new systems to understand the fundamentals. And just around that time, Julius and others in MIT, they start thinking maybe we can give the iGEMers, the parts so they can start, building biology systems. So in a way, we can better engineer and understand these systems. So this has been really opening up the field of synthetic biology in the last 20 years. And many people really think about biology almost like receptacles, right? So very often, we start thinking about parts. We think about these are essentially transistors. You can put them together to build more complex chips. So I just want to take a moment to remind you, we have gone a long way in the last 80 years in the chip business, right? So in 1948, the first transistor was built, but it took 10 years to actually build the first integrated circuits. Only 5 parts, okay? Only 5 transistors. And it takes another 20 years to have the first Apple computer. But today, in your pocket, you have about 20 billion transistors in your iPhone, okay? And if you're really thinking about scales, you think about, we're sitting here, right? So today, we are basically 99% of the genetic engineering we are doing is less than 50,000 base pair of DNA, and only a handful of parts. And this is equivalent to the first integrated circuit in 1958. So if we really think about building microbial genome like Jason was talking about, I just want to remind you there's a huge gap in between, right? So we're talking about million base pair DNA needed, over thousands of parts you need to put together and understand them and try to build a new system. And so you basically need to make a leap of 100-fold increase. But if you're being so ambitious, like Jason and others, so you may ask the question, what if we can really get there? If we can build genomes, what can we do with it? Right? So actually, since early 2000s, we are making so much progress in this field of writing the genome. So it's all started with polyvirus and T7H. These are virus DNAs, and the T7 was pioneered by Julius, and then move on to bacterial genome, exampled by the work from J. Craig Venter Institute and Jason Chang's lab in LMB Cambridge. And I coordinate the first synthetic eukaryote genome, AC2, with Jay Booker. So we have been building eukaryote genomes. So we now complete all the synthetic chromosomes in yeast. We're just well on our way to merge into the first eukaryote genome, AC2. But there are already movement in the space to start making much more complex genomes, such as the plants, the humans. So what if you can write genome that would really open up all kinds of applications, ranging from medical, food security, industrial, environmental. And these are the biggest challenge this planet is facing. So how do we get there? So we really need to embrace the engineering technologies. So for instance, genome design cannot be done by hand, right? So when you have only a handful of parts, you can design by human experts. But to scale up to genome size, you need much more complex computer-assisted designers. And then we need to be able to embrace modeling and prediction so we can have an idea of the predicted phenotype before you even build them, right? So we need to embrace the design with manufacture. Like Jason said, if you cannot manufacture the DNA, what's the point of design? And finally, we need to think about optimization of manufacture and using robotics, and then eventually using AI to learn what's the outcome and go through the next round of iteration. I'm going to show you some of these examples. How do we engineer the genome at scale? So the first thing I like to do is show you a bit of the design we have done in the past 10, 20 years. So this is the key technology one, is generative design of synthetic genomes. So I'm not going to talk you through all these design principles, but essentially, you can really think in high level, in human language, what you like to do to the genome. For instance, remove all the redundancies, embed, codons, and add the log species downstream to every gene. You can describe these really simple rules in just a few sentences. But this represents thousands of edits per genome or per chromosome. It's very difficult to do it by hand. So essentially, what we have done is we take one of the genome designers, feed it with the design principles, and then automatically generate the synthetic genome in base pair resolution. So what you're looking here is chromosome 5. So this is 500,000 base pair in front of you. And then now the software suck up the design principles and generate the design in base pair resolution. And it's done in seconds. And And the next part is how do you really scale up the assembly, right? So in the past, we demonstrated you can build this synthetic DNA in vitro up to 30,000 base pair, and then you use the wild type chromosome as a scaffold, right? So you can swap in 30,000 base pair chunk by chunk. And this is essentially of taking a boot chapter, then you replace one paragraph by one paragraph, right? And then you use the micro system as a proofreader of your code, right? And this has been very efficient. But then we are thinking about, can we now take advantage of the fact yeast has two mating types? They can mate, right? So using mating, you can very quickly, convergently, assemble the DNA. And this will really scale up the assembly from 100,000 base pair to 1 million base pair, okay? And then as an engineer by training, I was thinking, maybe we can automate some of this process, right? Because human is very good at design and analyzing, but we're not really good at moving liquids, right? So this should really be a manufacturing problem. So I and others were thinking about using the automation or the genome foundry to automate some of the DNA assembly process. And finally, we are going to measure the phenotype, but from a multi-dimensional manner, right? So we really developed technology to look at DNA, RNA, protein metabolite genome-wide. So this allows us to pinpoint any dysregulation due to design. So I hope you believe me, we now really have the technology to design, manufacture, and characterize synthetic genome at scale. And over the last 10 years, we have built some of the chromosomes, some eukaryote chromosomes, I should say, one at a time. So we built chromosome 2, which is the first synthetic eukaryote chromosome in Europe. And chromosome 7, that's the largest one. And then the tRNA chromosome, which is a complete de novo chromosome, meaning there's no natural template. You have all the degree of freedoms to put a chromosome together. And these are all published. I'm not going to tell you through all the details, but I'd like to now lead you to the next key technology is we are not just mimicking engineering, right? So we are biologists. We embrace the concept of evolution. So the next key technology is, can we really coupling engineering with evolution? So we put in over 6,000 LOXP sites across the entire genome. And as you know, Cree would induce pairwise interaction of LOXP recombination. So the idea is we're not building just one synthetic yeast genome, but at the induction of Cree, you can generate billions and tens of billions of combination, and then you can select for the phenotype. And this is kind of crazy idea for engineers, right? Because you would not imagine someone build billions of airplanes and see which one is still fly in the air. But that's the power of biology. So by doing so, I can show you some of the applications. So the first one is we demonstrate by turning on the Cree, the yeast can transform into billions of combinations, and you've got really novel genotypes. So you can really, on demand, turn on the genotype diversification program. And then you can select the guys which can tolerate stress. And then you can also use that to improve the metabolic engineering. So you can evolve the strain which produce much better natural products. And finally, we can use it to minimize the genome. Right? That's the holy grail of biology, is figure out what's the minimum set of components which are needed for sustained life. So I have been telling you about the work we have been doing in yeast. Now I want to tell you a little bit about the other things we are doing out of the yeast, right? Because I'm a firm believer, when you build a hammer, everything looks like a nail, right? So the first thing we are doing is looking into the antimicrobial resistance space, right? So this is a prediction by UK government. By 2050, the death toll per year would be about 10 million people. That means 3 seconds per person would be dead due to antimicrobial resistance. So what we have been doing is looking at the nature as a source of inspiration. So we look at the phage because they naturally attack bacteria. So we are able to pick and choose modules from different phage and put them together to build new therapeutics. We just got started with a new ARIA project, and this really allows us to think about, can we engineer plants, in particular potatoes? And so the idea is, can we build a synthetic chromosome, put that into potatoes, so we can plug and play different traits? And this is to address some of the climate challenges for the crops. And we have been working with Cancer Research UK. So the idea is we have sequenced a lot of prostate cancer patients, and we identified regions of the chromosome which had amplification. Now we are building synthetic human chromosomes to identify the causal mutations. And so hopefully we can develop gene therapies. Okay, so far I tell you we can scale up about 100-fold from the current practice of genetic engineering to microbial genome engineering. So that's mega-based DNA synthesis. But if you really want to push the envelope a bit further to look at much more complex genomes, we're thinking about giga-based DNA synthesis, right? So that's 1,000-fold leap we need to take. So I'm not going to tell you too much about this, but we have been working with Jason's lab and Tom Ali's lab and Joey Zhang from Kent, and we are going to take the first step into making the synthetic human genomes. And more recently, my group has been collaborating with Tom Ali's group and Francisca Trioni's group in Imperial, funded by Zuckerberg. The idea is, can we build a pipeline which really streamline the production of synthetic chromosomes up to gigabase scale and test the phenotype and the stress induced to the cell. But we cannot just do this. We just think about responsibilities, right? So I think over the last 2 days, you have been in these responsibility meetings. So I would say, safety is important. Safety is not a constraint. Safety is the hallmark of maturity of engineering. If you look at other types of engineering around us, I would argue by embedding safety, that really help people to uptake the technology, right? Looking at the cars, you have so many orthogonal systems which make sure it's secured. So we have been building the technology to allow us to technologically embed what we call the safety switches. So the idea is we can target independent essential cellular process. They don't compromise the growth of the cell, and they're orthogonal to each other. If one fails, the other one can kick in. And they can work independently or collectively. And finally, these are universal mechanisms you can transform virtually to all forms of life. And how much it costs, I will tell you, it's very efficient. So with these switches, we can go down to 10 to minus 14 escaping rate. That means if one of these 1,000-liter fermenter break, you will not get a single isolate. And that costs about 17 cents US dollars to contain it. Okay, so I'm going to finish by saying, if a biologist cannot fix the radio, maybe he or she can build a genome responsibly. Thank you.
Thank you, Patrick. The next speaker is Julius Fredens from the National University of Singapore.
Good morning. Thanks for joining the main stage. I'm bringing a story from my lab in Singapore. And it's embarrassing, but I don't remember my title, so I hope it comes up on the slides soon. It's called Bridging Continuous and Discrete Evolution of Large Gene Clusters. Is this the clicker? Yeah? Okay. I got a big red button here to play around with. Thank you. I was a molecular biologist until I joined iGEM in 2009, and then overnight I became a synthetic biologist. And it's really exciting to be back here and see how iGEM has evolved over time and see how the research has evolved over time. So back in 2009, most of the research we did, most of the things we were building were a few components that had to play together. At the time, probably exactly at the time of iGEM, I believe Craig Venter must have submitted his manuscript for the first cell with a synthetic genome. It was a big deal back then. And And since then, I was fortunate enough to be a part of Jason Chin's work on synthetic E. coli genomes with compressed genetic codes. We have heard Patrick and Matthew are working on synthetic chromosomes in yeast. So we are clearly scaling. And now we have a session here at SB8 called Synthetic Cells. Casually, it's even plural. So things are really scaling quickly here. And when we start scaling our synthetic biology, we also need to be able to scale the tools that we need to refine them and get them to work well. That's what I want to tell you about here. So we need methods so we can start in a targeted way to evolve very large constructs, protein complexes, metabolic networks, even just single massive proteins. These are entities where all the different components or the entire protein, they have to co-evolve. We can't just take one piece at a time. We need them all to evolve simultaneously to refine their activity. So we set out in my lab to try to do this. I have a very good PhD student, Chang, who was keen to try to solve this problem. There has been some fantastic progress over the last few years, including from Jason, on orthogonal replication. That's certainly a strong method that can help us to reach the first, sorry, there's a laser here, to reach the first, there's no laser. To reach the first of the goals up here, accelerated evolution of large gene clusters. We're not talking gene of interest, we're talking gene cluster of interest, okay? GCOI. The only thing we wanted to add to it is we would like to be able to kick out the cells, so we are sure that it is only our GCOI cluster that is evolving, not the cell itself. That's a whole other topic. So my student here envisioned an idea. We tried to take a phage and get the phage to do the work, but at a large scale. And we jumped on the T7 phage because it's a pretty aggressive, pretty efficient phage. It has a genome of 40 kb, so it's manageable when we have to synthesize and handle the DNA itself. It has a latent period of 11 minutes from it enters the cell and until it is bursting the cell open and taking about 180 copies of itself out of the cell. Very, very briefly, the DNA goes in, it is transcribing and translating into a T7 DNA polymerase and RNA polymerase. They are replicating the genome and transcribing all the components needed to make the new small phage particles that are coming out. A nifty feature of T7 is that it is easily confused. If we introduce a small piece of DNA, it's a little plasmid, but we call it a phagemid. It contains 2 short sequences of T7 DNA. This plasmid can go into the replication system. The phage cannot figure out how to distinguish its own genome from this plasmid, and it even starts putting it into its own particles and kicking them out of the cell. So now we're ending up with a mixed population where some of these small phages, they don't contain their own genome, they contain the DNA I have put in here. And when they meet a new E. coli cell, they are injecting it, and it is recircularizing and now being maintained as a plasmid in the new host. So this is a nice little mix between phage and host DNA we're looking at. So the idea would be, can we get this little phagemid to transfer the genes we're interested in? We can handle them somewhere between 3 kb and 40 kb, so it's quite versatile actually. The challenge now is we would like this to be continuous, and the phage is terribly lytic, so there's no continuation in this. So how do we solve that? Well, first of all, we were looking at biocontainment. So if we take the DNA polymerase out of the phage and we put it on a plasmid and we supply it in trans in the host, then we have complete control of the phage itself. You see here the process is exactly the same as before, except that the phage has been slightly disabled. It is not providing its own polymerase anymore. We started looking at making really, really error-prone versions of the DNA polymerase because this is what drives accelerated evolution. And we could take a number of known mutations from DNA polymerase, both from the T7 DNA polymerase, but also from other polymerases. We could just look up on Google. We could transfer the positions in other polymerases and put them in. And we got a whole spectrum of different polymerases, as you see here, that all have pretty good error rates. And then we tried to boost it a little further by adding a deaminase structure to the end of the polymerase. So the polymerase is tagging along and making its usual errors. And then behind it is hanging a deaminase that pokes into the DNA and makes further modifications to it. And when we combine all this, we're getting to versions 8 and 9 here that can give us an error rate of about 1 in 25,000, and at the same time creating a pretty equal mutation spectrum with a deaminase that is turning both A and C into G and T. Now, something fantastic happened at this point, and maybe it's not surprising, we just didn't think it through. When the polymerase is error-prone, the phage starts struggling a little bit. So when you see it up here, if you look at the very left, we see that an E. coli culture infecting by a T7 phage with a wild-type polymerase, the culture collapses. It's almost dose-independent. As soon as there is a little bit of phage, the phage is replicating enough that it is killing the cells. We're looking at the left part of the lower figure here. We have exponential replication of the phage. But when we're introducing an error-prone polymerase, then the picture changes a lot. Suddenly, the phage is not able to sustain its own replication in an exponential way, and we start getting an infection cycle that is much, much more dose-dependent. And why is this interesting? That's interesting because now we can start creating a cycle with our lytic phage where we can use a high MOI, that's a high concentration of phage, to kickstart a lysis process where DNA is it's taken out, replicating, an error-prone replication, replicating quickly, taken out of the cell, and the culture collapses. And then we can move it, and now we have a low concentration of phage, and now it's just transduction into cells, and we can both insert our plasmids, phagemids, and we can select for the ones that are performing well. So this is creating a cycle for evolution that is really compressing the time it takes, and is still compatible with large DNA. So here I'm showing you, we put together, this is a pretty random yeast chromosome, we had to fill something in, and it had to be cheap enough to do quickly. So we put in 39 kb of yeast DNA into a phagemid, and we can show how this can be transferred through the replication process. We get a pretty neat mutation spectrum across the entire section. And as you see to the right, after one cycle of all this, there is barely any truncation of the DNA. So it's a pretty stable system, even when we're working with large DNA. So we set out to use this for something. You have to do that, right? We set out to use it, and we found a pathway where you can transform the plastic polymer PET. You can degrade it down with PETase and METase into TPA and ethylene glycol. And ethylene glycol can then be coupled to the metabolism of the cell and be turned into energy and biomass. It's actually a very simple process. It's dependent on 5 enzymes, as you see up here. So we took these 5 enzymes, put them together on a phagemid, and started evolving them. We went through 5 cycles. We did semi-relaxed selection. That means that we were helping the cell with a little bit of glucose in the beginning, just to let it live, right? And then we slowly removed the glucose, so the cell had to get better and better at just living off of its ethylene glycol as its sole carbon source. We did 2 different experiments here. We did the traditional adaptive laboratory evolution, or ALE. That is that you take your cell and you just let it grow in the lab for a long time, and you let the cell figure out how to solve the problems. The cell figures it out. The cell is actually pretty good at figuring out how to grow better, or in some other way, how to avoid the selection. So what we see here is that we get improved growth of these cells, but when we then go back and we sequence the genes that we want to evolve, we found out of 32 clones, none of them had actually accumulated any mutations. It's something else in the cell that mutates in order to circumvent the selection pressure. On the other hand, when we're using our method, lyse, then we get a very sustained growth, and all of the cells that are improved in their growth rate, they all have mutations in the genes we're interested in, or in the phage mid in general. And when we go back and we take these mutations, I would love to use the laser pointer, but follow me to the right in the single mutations into new host. When we took these single point mutations that we found in our screen and we moved them into fresh phage mids in fresh cells, they were imitating the same amount of improvement. So we know by this method that when we see improvement, it is sitting exactly in the genes we want, not somewhere else in the cell. And the best clones we could pull out of this, they increased their endpoint biomass by 51%. So they got 51% better at taking up this plastic degradation product and turning it into bacterial biomass. So with this in hand, we feel pretty confident that it's working. So my PhD student has now gone to Tobias Erb's lab at Max Planck. His lab is working with the beta-hydroxyaspartate cycle, which is thought to be a very, very efficient way of converting glyoxylate into oxaloacetate. And most of us will probably say, well, that sounds exciting, but why are we doing that? The point is that glyoxylate is one of the products in photorespiration, while oxaloacetate is one of the ones that is used in the metabolism that is then turned into biomass or energy. So by making a very efficient combination, you can effectively make photosynthesis much more efficient for the plant. So they came up with this. It's not a synthetic pathway, it's just a very recently described pathway that is only consisting of 5 enzymes that are turning it through 4 different intermediates. No carbon loss, very little energy usage along the way. And when they start using our method to evolve this pathway, we see a fantastic improvement over very, very few cycles. And we also see you can evolve this in one host that is particularly good for the topic. And when they have evolved to a certain level where things are getting better, you can move it to another strain very easily and start continuing there. So you have this mobility between strains that are fitting your purpose. That's everything I wanted to say. This was from my lab in Singapore, mainly driven by my PhD student, Xu Jianong, but I have highlighted the other people that are involved. Thank you very much for your attention.
Thank you, Julius. Our next speaker, the Petra from the Max Planck Institute of Biochemistry.
Yeah, so do I have to click or? No, apparently not. Here we go. Yeah, so I'm a bit of an outlier here because I'm obviously here in a genome fan club, and I'm not going to talk about genomes at all. But Patrick has already given a perfect introduction into my approach with the question, how would a biologist fix a radio? Well, I'm not a biologist, I'm a physicist. And a physicist would fix a radio slightly differently. He or she would probably look out for the smallest possible radio that they could find and then try to understand what are the really essential parts that make that thing work. And that's our approach to synthetic cells. We do not want to deconstruct and reduce. We want to understand what does it actually take to be a cell. And unfortunately, as you all know, we might find the most simple radio in any of our museums, but to find the simplest cell, which has probably been there 3.5 billion of years ago, there's no way to find it. So the only way we can do is to try and build it and try to understand or define from scratch what do we actually expect from a cell, from a living system. So we have to do some thinking before we start. And for a physicist, the most amazing thing that a living system can do is to be able to self-organize in space and time. As you might know, this is not only possible for living systems, also chemical systems, if they are complex enough, can do that. But if you really want to understand what it takes to be able to sort molecules in a compartment, and this is what cells are doing all the time, although all the molecules are diffusing, a cell is still able, by the smart use of energy, to sort some molecules to one side and other molecules to the other side, and that's something we need to understand how that works, otherwise we cannot build it. And then the whole fueling of this, the whole field of metabolism, of course, is a black box for people that are coming from physics that we leave to the biochemists and molecular biologists. But there's one feature about cells that we as physicists really love, because it has to do with physical forces, and it can be witnessed under our favorite observation machine, the microscope. And this is cell division. Every cell, and that's a very obvious quote, is able to divide itself and replicate. And this is something, if you could manage to build a system that is able to self-organize in space and time such that it can divide and you can see it, that would be quite something. It would not be a living system. That would take another couple of features, but it would be a first important step. And that's what my lab is working on. And we're a bit cheating, of course, because you have to have functional elements to start with. And so we are working with proteins. And this is, of course, something that my Origin of Life colleagues would always blame us for, because, of course, proteins haven't been there from start. They probably arrived a bit later after the RNA world was through, who knows. But the good thing about proteins is they have assigned functions, and we know more or less what to expect from a protein. So our approach is to be able to find the smallest number of proteins, a manageable number of proteins, that we would need to put together in a compartment that could basically build something like a cell. And this compartment we built from phospholipid membranes, because every cell on Earth has membranes around. So our approach is to deliver a small set of functional elements in membrane vesicles and see whether we can reconstitute spatiotemporal self-organization and bring these things to divide the compartment. So how to do it? Of course, you want to take some, ideas from how nature is doing it, and there's one obvious thing. Every living organism does self-division in a different way, has different machineries, but there's one recurrent theme that many of the cells are actually using, and this is the contractile division ring. So if you look under the microscope, you find a considerable number of cell types that assemble something like a contractile cytoskeleton into a ring, and it looks under the microscope as if this ring would constrict, and this would basically give you the forces that it takes to divide that thing. For example, Bala Subramaniam's group has done that very beautifully with actin rings that were purified from ghost cells, where you can very nicely see how myosin motors are constricting them. That seems like an approach that we could take. Same with E. coli. E. coli also has the set ring, and if you look at them under the microscope, it also looks a bit like my little cartoon up there. So why not trying that? We started with actin rings because they were large and they were contractile, and you could actually see in a single molecule experiment that indeed if you attach actin filaments to membranes and throw myosin motors on them, the little red dot is the myosin, you can actually see how the actin is constricted. And we managed to get beautiful actin rings in our vesicles. By the way, there's a scale bar missing. These are very large, so they're about 20 microns in diameter. The problem comes if you now take that myosin in these cells with the beautiful actin rings, because you do see that myosin constricts the actin. You can see it in the lower, for some reason I don't have a pointer here, but you can see it in the lower row here. The rings do not look quite as nice as up there because myosin needs a little bit different soup of ions. You can see that these rings constrict nevertheless, but they do not constrict as if we had imagined it in a little cartoon. What happens is that the actin just snaps off in a little blob at the pole of the vesicle, because what we did not think of course, the actin can slide in the membrane. The membrane is a fluid environment. And if you have something that is attached to a membrane molecule, a lipid, then it can be moved around. And if that would constrict, it would not bother the vesicle. It would just constrict and build a little blob. Well, that's not the approach we want to take. So we somehow have to keep our rings in place. And the solution to this comes from E. coli, because E. coli is really has engineered a super cool protein system that is there for keeping this division ring, the set ring in E. coli, in place. This is the min proteins, and the min proteins are a self-organizing system of only 2 proteins that need ATP to execute oscillations between the 2 poles. And these oscillations between the 2 poles are there to position the set FDZ and all the additional set ring molecules to the center of the cell. If we reconstitute FDZ, not ANKTID anymore, but FDZ to our vesicles, it looks not quite as nice. As you can see, there's not a single ring yet. It forms more like a filamentous meshwork. But as soon as you add the min oscillations in our vesicles, you can see that this meshwork is basically pushed together to the center of the vesicle and forms a beautiful ring there. Now, does that ring also constrict? That's a little bit tougher, because apparently constricting membranes is not quite as easy for FDZ as it is for ectomycin, because FDZ does not have a motor protein. Nevertheless, if we express our FDZ and our min proteins in vitro in our vesicles, you can see in this movie very beautifully that over time, the FDZ is not only pushed to the middle, but also starting to constrict away from the spherical state. And we very recently also found another part of our multi-protein FDZ ring that helps constricting even further. This is FDSN. This is, if you have FDZ on a surface, you can see that it makes constrict. And if you add it then to our vesicles, you can see that the constriction goes all the way almost to the full scission. So this is a set of 6 proteins. So that's pretty good already. And now we see where we can go from there. But I would like to take the last 5 minutes to show you something about serendipitous results, which might be even more important for our overall goal of constructing cells from scratch. Because we found that these min proteins, the beautiful oscillating proteins that are there in bacterial cells to position the set ring to the middle, do very amazing things in our synthetic cells. Because they do not only push a ring, they also bring slightly deflated vesicles to bounce around and even do a scission without any filamentous cytoskeletal proteins. Just by a massive binding to the membrane, they're pushing the membrane outwards. And, oh, sorry about that, I wanted to go back to this. Oh, I hate this. I wanted to show you the other movie. Yeah, this one. Here you can see what these min proteins are doing with deformable membranes. If you have little green FDZ rings, they build almost like Jeff Koons balloons out of them. In this set, we have 3 proteins, 3 different proteins, and they're doing all kinds of fancy things. Things they are never doing in a bacterial cell, because a bacterial cell has a very stiff cell wall. Even more, these min proteins do not only position FDZ on a membrane, they position nearly everything that can diffuse in the membrane. And if I say everything, I mean even non-biological molecules. So as soon as you have a molecule that is diffusing on a membrane, like it's anchored to the membrane, you can basically push it away and form patterns of it with the min proteins. And this is by a process called diffusophoresis. It's basically meaning that the massive binding of these min proteins to the membrane, as you can see in the cartoon in the left corner, you have something like a diffusional ratchet on the membrane. So the massive binding of min proteins doesn't allow other proteins on the membrane to diffuse in the direction where the mins are coming from. So they can only diffuse in the other direction. And by this, you can basically push away molecules from the membrane and create patterns and void zones on any biological membranes. And that also works in cells. We've checked it. And last but not least, you can take the min proteins to execute active transport of vesicles, as you only would see it with motor proteins usually in eukaryotic cells. So what you're seeing here is a vesicle that's about 10 micron in size, and is basically a membrane on the bottom, and the min proteins are forming patterns on this membrane, and because they're binding to the vesicle, they will deform the vesicle. And by the deformation of the vesicle, the min pattern is deformed too, and so you have a chemomechanical cycle going on. So what we see here is an active transport fueled by ATP, which has nothing to do with motors. Okay, so since these min proteins are actually doing a lot of cool stuff, we figured that it might be nice to engineer something that is similar to the min proteins, but simpler. And we have actually found that you can take one of these, min E, and dissect it in different parts, and can actually learn from dissecting the different functional parts of the molecule what are the most important motifs of this protein to be able to form these exciting patterns and functions. And we found that you can actually chop away a lot of the functional elements, and you can actually remain with a little helix, a peptide helix that is supposed to talk to the other min protein. And you can either attach the peptide helix to the membrane, or you can dimerize it, and both give you a functional self-organizing protein. And of course, this leaves us with the fully new perspective of building proteins from scratch. And everybody knows there's a lot of excitement in the air about protein design. We can now go ahead and take what we've learned about our pattern-forming proteins to feed it in the computer, that the computer design proteins for us. And we've recently succeeded in designing a min DE protein that you could see in the last slide with machine learning. Petra is the student who actually did that. So we took the protein, the machine-engineered protein, and could basically screen for it in our synthetic cells, and then could take it, the one that was forming patterns, that were forming oscillations, and put it back in bacterial cells and could rescue the phenotype. So it really is also in vivo functional. And this gives us a new angle to protein design because, of course, like everybody who works with proteins, we want to embrace it. But we want to not only design proteins for in vivo applications, we want to design proteins for our in vitro minimal cells because we believe that we probably need different molecules. We may need simpler molecules, and we can potentially even come up with something like a toolbox of parts for synthetic cells. And in the long run, I do think that this is a quite interesting engineering aspect to biology. It's still a long way to go to design something that is living, but to design some important features of life, I think this is a very good approach. And with this, I would like to thank all my co-workers and collaborators and you for your attention.
Thank you, Petra. Our final speaker, Kate from the University of Minnesota.
Thank you. Thank you so much. I have a greatest pleasure to be the last one in the session, so I get to summarize everything, I get to put it in context, and I get to basically stand on the shoulders of the four giants that spoke before me. And what I want to talk about is generally synthetic cells for useful practical applications. Not that my lab does anything useful or practical, but just kind of putting it in context. Why are we doing it? What do we want to get out of it? So starting with the context, some of you probably are sitting here in the audience, if you're new to the field of synthetic cell engineering, you're probably wondering, why the hell are we doing this? Why are we here? When you think about nature, nature is so incredibly diverse. There is so many different types of form and function in nature. So why do we bother making synthetic cells? Why do we even start from scratch? And the reason for that is those of you who are chemists like me understand that there is only one type of life on a chemical level, on a biochemical level. All of life on Earth is identical. There is all of the same metabolism, only 22 proteinogenic amino acids, only one type of nucleic acids. It's all at the same time incredibly simple and incredibly complex. On the chemical level, it's simple. We don't have that much chemical diversity. But on the structural level, it's incredibly complex because we don't even have a full ingredient list of any single complex living natural cell. So what we want to do in our field is we want to make synthetic cells basically so we can fix the radius, so we can understand how cells work. And as Richard Feynman said it the best, if we can build it, then we can actually understand it. We'll be able to say we actually understand living cells. So what my lab is doing is we take liposomes, phospholipid liposomes, we make them look sort of like cells. So we put channels in a membrane so we can put things in from the environment, so we can remove waste, and then inside our synthetic cells we put cell-free protein expression system. And that's it. That's the simplest, most basic cell. And what we do is we build genomes that are incredibly simple. So our genomes are nowhere near on the level of the genomes that Patrick or Jason were talking about. We're doing something very, very basic, but the one advantage we do have is we know exactly where every gene of our genome goes and what it does. And just to put it in context, if you look at the complexity of genomes that we can do in this very bottom-up, simplest minimal cell, we are an order of magnitude below the complexity of even the simplest known living cell. But the advantage we do have is that every gene in my genome is known, has a known function. I know where it goes and what it does. The really fun part of the synthetic cell engineering is building the cytoplasm, building what goes inside, and building the whole metabolism. So the metabolism, the cytoplasm of my synthetic cell is a cell-free protein expression system. So it's basically a lysate of E. coli, most of the time E. coli, that has all of the components that you need in order to make a protein. And that's what we actually play with. That's actually the kind of a chemical tool set that we can work with. And I'm just going to show you the kind of a rationale for the whole field. So what we're doing is we're calling it bottom-up. The bottom, the molecules, non-living molecules that we're putting together into a cell that is right now not yet living, but we're getting to the point where the complexity is starting to approach the reactions that you would expect out of a living cell. The first half of the sessions today were this so-called top-down approach, where we take a complex living cell and make it simpler, simplify it to the point where we can start understanding everything. And the goal, right now, the biggest goal of our field, the sort of a holy grail, is for those two approaches to meet. We want to be able to take the approach that my lab is taking and that Petra is taking of putting together molecules to build up the complexity and combine it with the approach of the people that take the complex living organisms, make it simpler to understand it. In the end, we're hopefully going to meet one day and we will have a fully controllable, fully understandable synthetic cell. So couple examples of what this very, very simple non-living synthetic cell can do. We can make genetic circuits. We can create genetic circuits inside a synthetic cell and between two different populations of synthetic cells. This is a very simple example of one of the works from my lab where we show that this is dead as a doornail. This is not a living cell, but it can create this kind of emergent complexity where you can create genetic circuits using proteins, RNA, all the normal components, but you actually have a full control over what you're engineering. If we want to build more lifelike functions too, we can then, I apologize, the quality of the imaging that we can do in my lab does not match Petra's imaging. I'm really glad that I'm speaking after her, not before, because you forget about her imaging. I can just show you those simple little liposomes, but this is what it looks like. Two different populations of synthetic cells. When I'm imaging them, they just look like those little dots under the microscope. This is two populations of synthetic cells. Each of them expresses a different color of a fluorescent protein. Simple system. If these were bacteria, you would say, what's the big deal? But these are completely chemically defined synthetic cells, so they have a pure system as their cytoplasm. We know exactly every single molecule, and here's the lifelike, very basic but lifelike system that we can engineer. If we put antibiotic resistance into one of those populations and we add antibiotic to the whole population, the ones without the resistance I can't say they get dead because they're already dead, but they get deader. They basically stop producing proteins. Again, what's the big deal? This is not, bacteria do it all the time. But this is a fully chemically defined system. I can give you a full ingredient list of every single compound in this sample. So this shows you that we're getting to the point where our system starts sort of looking, if you squint right, it's starting to have the complexity of a lifelike cell. So why are we doing it? What's the rationale of it? Some of the things that my lab is really interested in is using synthetic cells to understand natural biology, to reconstitute some of those complex biological processes, not just antibiotic resistance, but other complex biological processes that cells naturally do in this controlled environment. And by doing that, we can make better tools for bioengineering. What you need to build a synthetic cell is also a great tool if you want to engineer both synthetic and living cells. We obviously have to pay the bills, so we're interested in biomedical applications, we're interested in making drugs, and we're interested in using synthetic cells as a platform to make biologically active compounds and proteins. That also translates into metabolic engineering. We can use synthetic cells to build pathways. We can use synthetic cells for really fun applications. For example, if you take a synthetic cell system, you can lyophilize it and store it at room temperature for weeks to years, and that's really great if you want to send Elon Musk to Mars and have him stay there. He's going to have to make medicine at some point. Astropharmacy is one of the applications, and we have demonstrated that those systems do work under spaceflight conditions. Petra mentioned the origin of life. Those systems are great for reconstituting the early lifelike processes. For example, we tried to reconstitute the earliest evolution of the RNA world on Mars using synthetic cells as well. Making new materials and making biocomputing solutions, these are the things that you can do in synthetic cells with much greater flexibility and kind of a fidelity and yield of production than you could do in natural cells, just because you have this more control over the chemistry. Okay, why are we doing it? Why bother? Well, we bother because we have to. There is no other way. Right now, when you think about the current world's economy, everything that surrounds you is coming from oil. There's probably the usual suspects. When you think of things that we get from oil and petrochemicals, it's the fuels, it's the plastics. But it's also everything else around you. Everything that you're wearing, all your electronics, all the drugs you're taking, and all the food you're eating. We could not feed the current population of the world without petrochemically derived fertilizers. So this is the current state of the world, and we cannot maintain that. That's pretty clear. We cannot keep running our economy on oil. So we have those amazing renewable energy sources. The problem with renewable energy sources is that that's exactly what it says on the box. It's a renewable energy source, not a renewable chemical source. We do not have right now the way to replace all of the petrochemicals that we are getting from oil. We do not have it at scale, but we know how to do it. We know what we need. We need a bioreactor. We can grow biomass, and if we grow biomass, we can then process that biomass into all the chemicals that our economy needs. So if we grow feedstock, and we grow it wherever it wants to grow, so native feedstocks in every part of the world, we can process that into chemicals. Now, how does synthetic cells come to this? Well, because those bioreactors have to run on something, and we want to make a case that the kind of a worldwide bioeconomy cannot be built just using natural cells. We need this full operational control over a living cell, and that is a synthetic cell. That's something that only a synthetic cell can give you. And when you think about building your bioreactors, if you compare what you can stuff into your bioreactor, natural bioproduction strains, natural cells, or synthetic cells, if you choose to use synthetic cells, they're going to be much safer. Patrick already touched on that, biocontainment and the ways to engineer synthetic bioproduction pathways. It's much easier if you have this fully controllable synthetic cell. Then it's also much faster because the timeline of prototyping metabolic engineering pathways in a synthetic cell, it's on the order of hours instead of on the order of days or weeks like you do in a natural cell. And that all translates to lower cost. We all love saving money, and really to scale it up into a worldwide biomanufacturing, we need to make it cheap and accessible everywhere and to everybody. And that's kind of the world today. Only about 10% of manufacturing, chemical manufacturing in the world is being done by biology. And that includes things like ethanol for fuel. So this is where we are today. Where we need to be to survive as a civilization is by 2050, people predicted that about 60% of chemical manufacturing will be done by biology. I'm hoping this is actually a pessimistic prediction. I'm hoping we're going to be much better than that. But again, what do synthetic cells have to do with it? Well, we hope that when the world really switches to bioeconomy, at least half, if not more, of the world's bioproduction will be made with synthetic cells, with those cells where we can completely control their chemical composition. And that's going to give us this flexibility and scalability of biomanufacturing. Okay, so I said we have to do that by 2050, and some of you are probably thinking, no way, this is absolutely impossible. So I want to give you one example of how impossible can become possible in science and engineering. I think 2050 is actually an absolutely realistic timeline, because when you think what happened 100 years ago, in 1903, the Wright brothers built basically a flying bicycle with wings. And 17 years later, we had something that if you look at it if you saw it in the sky today, you wouldn't even be surprised. It looks almost like a modern airplane. This is 17 years. This is the timeline from the introduction of the first Android phone till today. So you can see that the technology can really progress at amazing pace, at amazing scale, if we are properly motivated. And I think the survival of the civilization is a pretty good motivation. So I would say engineering biology to the point where we can really do bioeconomy with engineerable, controllable cells with this full operational control of a metabolism is absolutely doable within not just our lifetime, but within the next couple decades. And we absolutely have to do it. All of those applications that I was talking about, they're not just kind of a fun things to do because we can. They're absolutely essential for the survival of our way of life as we know it right now. And those all are made possible by our ability to engineer cells on this absolutely molecular scale, playing with biology, treating it as chemistry. And we're doing it as an international community. That's another really important thing, is we have to distribute the benefits of this technology really equally around the world, and we have to work on it together, because not a single lab or not even a single country will be able to make this reality, to make this dream reality. We have to do it as an international community, and we are doing it right now as an international community. We are part of this great Build-A-Cell community that brings together people from all over the world that are interested in different aspects of engineering synthetic cells at scale. We want to be able to say we're actually engineering cells to do all those useful things, practical things, and make it happen together as a community. And I'm really happy that this event is happening here at iGEM, because iGEM is one of those examples of how you can bring international community together and make them work together towards the same goal. And that's all what we're trying to do with synthetic cells. So I want to say thank you to all the people in my lab. I haven't really shown too many results from my lab, but there's amazing people that do believe in it, all the funding sources. And I do want to leave you with this countdown. This is a countdown to 2050 by seconds. So by the time this hits 0, the world bioeconomy has to run on synthetic cells. So that's the do-out. That's the task I'm leaving you with. And thank you very much.
Okay, thank you so much for amazing presentations in the first session. I think this should be the way we start the day every day, so with science and amazing scientific progress reported here. We'll open the floor for questions from the audience. If you have a question, please raise your hand, and microphone will magically come to you. Okay, that's good. But as a chair, I always want to exercise the privilege of asking the first question. So I'll ask the very first question for all the speakers today. In your view, where do you think your fields are actually heading? I mean, okay, following Kate's time scale, in 5 years, 10 years, 17 years, how the fields of synthetic genomics and synthetic cell will continue to evolve. If you can share your thoughts on this very quickly, I really appreciate that.
I absolutely agree with what Jason just said, right? So a lot of what we do today is try to define sensitive genotype, which is sequence, and then try to derive some kind of phenotype. But in biology, a lot of functions are not sequence defined, right? Such as centromeres in higher eukaryotes. So I think the next big thing is how do you synthesize function without, knowing the sequence, right? So I think that's the next big thing. And I'm always excited about the possibility to intersect synthetic biology or synthetic genomics with AI. So you have seen examples of you can now use large language models to generate, languages, or to give you semi-perfect, paragraph of what you detect. And OpenAI is a good example. You can use it to use ChatGPT to generate new paragraph which never exist. I think it's the same in biology, right? So you have seen EVO2 as an example. You can generate sensitive phage genomes, and how do you do that at scale? And I guess what I kind of touch on in my talk is how do you do it responsibly to safeguard the technology intersection. It's very important.
I feel Jason and Patrick has covered the synthetic genomics very thoroughly there with strategy as well. Maybe I'll move over to the synthetic cell part and wonder if we start getting to a point where we can build a small engine that can run on its own, and it's not a synthetic cell we're building, it's more of a production platform, and that will actually do the functions, as Kate also alludes to, the functions we're looking for. It's not necessarily a cell per se, because we already have those.
Yeah, so I'm a bit. I want to pour some water in the wine, because I think just on that part, on the functional part, we are a little bit blind still. I mean, if it comes to genomes and to how genomes translate into structure and eventually into function, I would say we are very much focused on the first part. And if you would have to define function as such, it would be very, very hard. This is also something where I'm a bit skeptical how fast our AI will move on that part, because we have a fantastic database of structures out there, right, which you can use to create new structures and devise binders based on very obvious structural fits. But function in the true sense is not yet a very well-defined aspect. I mean, I tried to show you that we cannot even predict function from the molecules we know already, because you expose them to a different environment and they show completely different function. So we cannot think one-to-one if it comes to function. We basically also have to explore the whole context, and that's going to take a lot of experimentation before you can even hope to make an engineering of function possible.
All of the above. We're going to reprogram the chemistry of life. We're going to build genomes at scale. We're going to predict function. And we're going to have a self-replicating synthetic cell that can reach the complexity that can realize all of the functions of living cells.
Okay, great. Thank you.
Excellent.
Now, questions from the audience. Yes, back there. Yes.
Hello. I have two questions, actually. The first question is for myself. Since now we are building new molecules and new chromosomes and even new cells, can we say we are generating new species? Yeah, that's the first question. The second question I want to ask on behalf of my student who is still in China. She asked me to ask a question for her, mainly to Professor Patrick Tsai, because she's working on the scramble system, and she's asking the recombination efficiency of the fragments between the LOXP sites. If she put a plasmid with LOXP sites in it, will the efficiency between the fragments between the locked P site the same as the ones in the chromosome? Thank you.
Thank you. Patrick, maybe the second question first.
Okay, I think the scramble system is quite efficient, and we don't really have the hard data on how far two lost species can recombine in the chromosome, but they seem to be, very efficient, and the distribution seems to be distance dependent. So the two lost species are closer to each other, more likely to recombine than the others, but in general, it's quite efficient and a very stochastic process. What's the first question?
The first question, anybody willing to answer? It's a very deep question. Are we creating species?
I want to add to that. I think we're not just making new species, we're making new domains of life. Because, for example, what Jason's doing, translating amino acids that nature never translated in the over 3 billion years of history. So we're definitely making new branches of the tree of life.
Okay. Yeah, and I agree. I think speciation is kind of arbitrary concept, and there are many ways to define speciation. And for instance, if you do microbiome research, you sequence the RDNA sequence of Jason Chang's and the normal E. coli, you say, oh, that's the same species. But they're fundamentally different, running off different genetic code. And like Jason said, some speciation concept also rely on mating. So if you cannot mate with the other, strain, that means you're a new species. But it's very diverse concept of, what's a species.
Okay, great. There's another question in the middle section right there. Please, thank you.
Hi, I guess I'm really curious, kind of jumping off of that. Kate, you had a slide talking about, okay, there's this top-down approach and then there's the bottom-up. And you were calling your cells dead, and Jason, your cells are alive. And I'm really curious where you think life becomes death and death becomes life, right? If you were to meet in the middle.
Well, I'm not sure whether I'm supposed to answer this, but I would like to get back to this question of what is death. I mean, I would say death is the ultimate definition of life, right? So if something cannot die, it cannot live. And our systems, the bottom-up systems, they are at the moment still, they are dying because of energy shortage or because, the machines malfunction. I guess everybody knows that every protein, every biomolecule will degrade at some point. And I actually think that we haven't fully defined death in on a molecular level so far, but I do think that this is probably something that we will learn to do as our systems become a bit more interesting. So the ultimate aim we want to achieve is to have a system that can evolve in the Darwinian sense, because this is the only very clear definition what distinguishes a living from a non-living system.
Quick one, yes.
I think definitions of life are so fuzzy that according to Jason's definition of life that he just gave, I'm dead because I cannot self-replicate. I need my husband to replicate. So that just shows you how messy that is. We really don't know.
Okay. Next question, please.
Hello. So my question is a little bit less philosophical than the other ones before. So I was wondering, many of you are aware of the recent paper from the Arc Institute where they used their genomic language models to design new genomes for phages and tested them experimentally. So while this is probably like a big breakthrough for AI to demonstrate its capabilities, I was wondering from your perspective of like actually building genomes in the lab and having like lots of experience with that, what do you think is still, or like where is the race? Is AI better at designing genomes or like that go beyond what we're seeing in nature, or is it our experimental methods that we can, for example, scramble the genome and explore different combinations of it?
I did not hear.
So how we can design genomes better? I mean, that question will be touched upon in the next session as well. So please come back for the next session. But Patrick.
I think if you're really thinking about large language models for genome design, I think we are making breakthroughs. You have mentioned Brian his work on generating the the phage genome and then demonstrate it's functional. I think that's a very major breakthrough. But how do you go beyond microbial genomes is a big question because, like I said, a lot of these are not sequence defined, right? So AI model is really good by reading books, right? Reading text and then generate new text. But when the semantics is beyond the text, and that makes it very difficult for the AI model to learn. And I think that's the big question here is how much they can really generate the sequence which can reconstitute the functions which are beyond the sequence.
Okay, great. I think we got time for 2 quick questions. Yes.
Yeah, hello, thank you very much. Really amazing talks. Just maybe considering that genome recoding and all this work in synthetic cells increasingly depend on like large-scale synthesis and high throughput analysis, how do you navigate from your perspective in the field this tension just beyond the safety aspects, this idea of accessibility and the demand of all this high-scale throughput with the urgent need to prevent the centralization of these developments in certain institutions, especially with this idea that you want to push a bioeconomy based on, for example, the use of synthetic cells and all that?
I can't hear the question.
Oh, yeah, that's better, or?
Yes, maybe we can take the question offline, if you don't mind, during the break.
Yeah, sure.
Okay, great. Maybe, okay, 2 quick ones. The back there, yes. The 2 hands, yes.
Thank you. Hey, I'm Basti from the MPI Marburg. I have a question for Kate. Thank you for pointing out that we need, like, until 2050 a different economy. I just wanted to challenge a bit with with synthetic cells. Like with synthetic cells, we have to compromise between all the parts. Why do we not just use like biosynthetic chemistry and only use enzyme cascades for your endeavor?
Because you have to make those enzymes somehow, and that always goes back to a living cell. That's why we want a living synthetic cell that's very controllable.
Okay, great. Yes. The last question, probably, on the front row, yes.
Okay, thank you guys for the advanced presentation. Your presentation have beneficial to me, so I would want to seek some suggestions. Okay, now we even able to synthesize a large-scale genome and even to synthesize the cells, new proteins, and we have produced many small molecules and natural products. But now we are facing a problem. We want trying to use like yeast to produce a peptide containing 10 to 15 amino acids, but we have facing many problems. We also trying to use the YQ yeast lookout to screen the protein teeth. And I want to know if there are some other methods where we can try.
Other methods for
producing the peptide in the yeast, especially the customized peptide.
Other peptide production methods is a question.
Put it right here.
Andrew, any way could try, or any other method we can try?
Maybe in general, genome redesign, any new ways on the horizon in your view? Great. I think this really serves as a nice transition to the topic we'll be touching upon in the next session, how we can improve the way we do bioengineering. So hopefully you can come back at 12:30 for the next session, and we will take a break for 1 hour from now on. But let's thank the speakers one more time.